I took some time ago in front of the United Nations headquarters in Geneva. I think this is the most important thing. Collegiality facilitates collaboration, and collaboration accelerates progress. So I see my aim here to cover this huge white spot which Russia is on the map of science and vision prosthetics. So I will try to translate the knowledge I've got here to my colleagues, and I will try to share with you some of our everyday life, because yesterday I got some questions about what we are really doing all that time. So I would like to have briefly our history, what are we doing, our experiments, is there any other effort in Russia, and if I have time I would like to share some ideas about what is my view of future prosthesis. So our laboratory was founded in 1970s on the base of Brain Research Institute. And in late 90s of 20th century, military forces had given our laboratory task of development of artificial retina using bacteriorhodopsin films. It was a really very interesting idea, and it is, I think, has its own future possible development, but funding was finished, and at that time we didn't have an opportunity to progress in this research. So after having done a review of worldwide experience in prosthesis, we found it most feasible to concentrate on cortical visual prosthesis. I will later talk why. So in 2003, we started to develop a feline behavioral model for initial evaluation of microelectrodes which are produced anywhere in the world. So I joined the team in 2005 and was able to participate in this experiment. The more we the more we were doing with cats, the more we realized that cat and possibly any other animal could not answer all the questions we have to that animal. What does it see? He can't speak, and we should point attention to a human. So we in 2008 started to concentrate on the possibility of implantation in humans. That's why we had to check all these issues of biocompatibility, stability, and safety. Sorry. I was asked yesterday how do we take care of cats. So I'm sorry, it is the only picture found of the cage because I thought it is so common. And when we were lucky to visit Key West this year, I saw that cats there are approximately the same kind of cages, and they are everywhere really, and they're very not so usual cats. They have 6 fingers, so it was quite a surprise for me. So here is our lab and my mentor, Boris Bazian. And these pictures are quite ancient because now we are not working with cats. So what did we do? We trained an animal to raise a paw, its paw, in answer to visual stimuli. We called it phosphine model. So it was a light source with several spots, and we trained it. Different animal had a different training time, so from 2 weeks to 3 weeks on average. And we checked the animal for possibility of implantation because of the character and how it behaved. After that, we implanted in V1 region electrodes. I will show what kind of electrodes we implanted, but also we implanted custom-made electrodes. And we were checking all kinds of parameters of stimulation, and we had an epicortical electrodes and intracortical electrodes, and that was the topic of my PhD thesis. And this is a brief characteristics of research animal group. So there were 13 animals participating in the study. Yellow are animals implanted with neuroconnex electrode arrays, and we divided animals in two groups. I think that's more easy. In two groups, and each group was checked for a different electrode diameter, and mostly we relied on the data gained in Dobell research and publications. So these are the arrays we implanted, and the results quite correlated with the results of the Dobell experiments. So we found out that intracortical stimulation is much safer than epicortical, and all the parameters of the stimuli. I hope we will have some questions after that. The further we went, the more clear we felt our directions of research. We had to reveal major biocompatibility factors in microelectrode cortical array implantation. We have to know and use the best knowledge in production of electrodes, but that is not the field of my expertise, so I certainly need your help here. So we need to find the best cover for the electrode, and make them as viable and stable and safe as we can. And with time, I hope this field will be less invasive and more safe. So this is the picture of custom-made electrodes we make. This is a Russian penny. In 2008, as I said, we switched to experiment on rats, and these are the results we presented at ARVO this year, so you can easily find them. And just a brief description of the experimental group. We took 8 rats and divided in 2 groups of different covering of the electrodes. And we used different microelectrode material, so different metals, steel, nichrome, tubingen, tungsten, and molybden. And of course, we found out that thin silicon carbide covering is much more effective, and we needed much lower thresholds to stimulate. Then standard physiologic varnish coating. So these are the slices we've got, hundreds of them, and thousands of morphology pictures. And Lisa Rudskova, my colleague, she's great at morphology, and all the questions about it may be addressed to her. We checked the gliosis and the quantity of fibroblasts, and we tried to find out what are the major factors influencing the stability, and found out that the proximity of a blood vessel is an extremely important factor that influences. So we think